Okay, so the point of this lesson is to chat through all the lessons about transport across membranes. So it is lessons 24 through 29, so it's 6 lessons. And what I find is often because you do it stretched out, you miss bits and connections and the sort of highlights of why each method is important, why each method is included. So we're going to run through all of them at once and just touch on the important differences as sort of like a summary lesson. Okay, so transport across membranes. So we are basically talking here into and out of the cell. Okay, there are ways that things are getting across membranes within the cells, and it's very similar processes, but what we're focusing on is entry and exit from the cell at this point. Okay, so your first big distinction is that you've got passive methods of transport and you've got active methods of transport. So a passive method of transport just happens. Nothing needs to increase energy, it's just a random motion sort of thing. And this is your diffusion, your facilitated diffusion, and your osmosis. Okay, so all of those happen sort of by accident. Okay, active transport processes, those require energy, they require ATP, or they're not going to happen. And that is, funnily enough, active transport. But it is also your bulk transport methods. So your endo and exocytosis. All of these three, exo, endo, and active transport, all require energy in the form of ATP in order to be able to happen. Okay, so we're going to run through each method one by one, highlighting bits and pieces on the way. So we've got 1, 2, 3, 4, 5 to get through, and we've got to keep an eye on time so I don't ramble on too much on one of them. Okay, so passive, active, diffusion, facilitated diffusion, osmosis, active transport, and bulk transport. Okay, we're going to work through them in that order, same order as in your course. So we shall start with diffusion. Now you've come across diffusion before, you've come across diffusion way earlier on in your schooling journey, and you're learning about these sorts of things. So it's important now that you step it up that little bit more, that we add a little bit more into it, and that we also understand how it relates to our other methods of transport. So diffusion, your 2 big points is that we're moving from a higher to lower concentration. So this is the same as saying down the concentration gradient. Okay, so if you say that the substance moves down the concentration gradient from a higher to a lower concentration, you're saying the same thing twice. Okay, so pick one or the other. Higher to lower concentration is nice and straightforward. It is a result of random movement. There is no control here, there is no intention, it is just random movement of molecules which causes diffusion. So higher to lower random movement, and here we are talking about uncharged, which is the same as saying non-polar molecules, okay, or very small molecules. Okay, so it's important. Uncharged, non-polar, because we're talking about movement through the phospholipid bilayer, through that hydrophobic core of fatty acid tails. Okay, very small molecules. The particular one here that we're talking about is water, but that is then more, we're going to talk about water in terms of osmosis, because water is not non-polar. Okay, water has a dipole, but it does still diffuse across the membrane. It can still get across that bilayer, and that's because water is so small. So, uncharged or very small molecules. And then you've got a few other things to bear in mind when it comes to diffusion. Okay, you've got to bear in mind the steepness of the concentration gradient. Steeper gradient, faster diffusion. Rather like a hill, rolling something down a hill. Steeper hill, faster rolling. So, if your concentration gradient is shallow, you don't get much diffusion. The steeper that concentration gradient gets, the more diffusion you're going to get. Okay, other factor, temperature. Okay. We've said that this is random movement. It's a random movement as a result of kinetic energy. Kinetic energy increases with temperature. So the more temperature you have, the more movement you have, the more movement you have, the more diffusion you have. So you don't have to get into the ins and outs of the chemical and physical level at this, okay? We're just understanding that it is random motion, and random motion increases as temperature increases. So that's sort of the two that are sort of always with diffusion, whether you're spraying deodorant in one corner of the room, that classic example. Okay, but when we're talking about moving in and out of cells, we've got to add to this. So we're going to add surface area. Surface area now is important, because if you've got a small surface that you're diffusing through, you can't get as much diffusion as if you've got a large surface. So increase your surface area, increase your diffusion. And the last one, which we've sort of already touched on, is the nature of the molecule. Because that nature of a molecule, because we're going across that membrane. Remember, we talked about it being non-polar. We talked about it being very small. That's the nature. So the less charge you have and the less size you have, the more likely you are to diffuse faster or diffuse at all. So you've got two that relate to diffusion in general and two that relate specifically to diffusion across a membrane. You've got 4 factors that are affecting diffusion at this point. Okay, so always higher to lower, concentration, random movement. It's a passive process. Now this is what you learned at IG. Okay, so let's now step this up to what about things when it doesn't work so well? We know a bit more about the structure of the cell membrane now. So let's step up our understanding of movement across it. And we're going to do that by looking at facilitating diffusion. Still diffusion, still passive. It's just that it is being facilitated. And to facilitate something is to help it. So all we're doing with facilitated diffusion is we're helping the diffusion. Okay, we're somehow helping these things to get across the membrane. So again, we have from higher to lower concentration. Again, we have random movement. This is still linked to the same story of kinetic energy. Again, we have these two. Our new thing is that we're going to be going through transport proteins. So we have this problem of getting across the cell membrane. certain nature of molecules can't get across. But if we have a transport protein, we have that opportunity to give them the help to diffuse through the membrane. So here we are talking in particular about large polar molecules or ions. Okay, so it could be large ions because ions, of course, are polar. Okay, so sucrose, for example, comes through by facilitated diffusion. Sucrose is large. Hydrogen ions come through by facilitated diffusion. They are polar. Okay, so we're getting bigger and more polar, not just one or the other or the both combined. So, new things to think about here is the fact that you now have channel proteins and you have carrier proteins. So in general, we say that facilitated diffusion happens through transport proteins. But then we have to be a little bit more specific. What are our transport proteins? They're either channels or carriers. So a channel protein allows free movement of ions. It can be gated or not, depending on the protein, depending on what its function is, depending on all those levels of specificness that we're not getting into at this stage. Okay, channel proteins can be gated or they cannot. Remember, you've got a vast number of different channel proteins because you have a vast number of different things that must come into and out of your cells. Carrier proteins, these ones are moving specific molecules. So a channel protein can be structured so that only certain molecules can get through, but it can also be a bit more general, whereas your carrier protein has to be specific. There is a specific type of carrier protein for a specific molecule, and that is because of how they work. Okay? It's the binding of whatever that's moving that induces a shape change. So if something different binds, it's not going to induce the same shape change. It's not going to work in the carrier protein. Okay, so again, this is a passive process. There's no ATP involved anyway. It is simply the binding that affects the bonds that are already in existence on the protein. Ring some bells. Okay, think about enzymes, think about inhibition. We've got a very similar process going on in this case. So, carrier proteins, we move through. Few other things that we want to bear in mind. Okay, so we talked about on the previous slide, we talked about concentration gradients, steepness. We're going to fit them in very small here. We talked about temperature. We talked about surface area. And we talked about nature of the molecule. So we've got all of these again, okay? It's all those same things. It's still diffusion, right? But now we've got an added level because it doesn't just happen randomly. This diffusion happens specifically. So it's happening through these proteins. So it is then also affected by the proteins. So 5 is going to be, it's going to be affected by the density of transport proteins. Are there a lot of transport proteins? Because it doesn't matter if our surface area is enormous if there's only 3 transport proteins. Enormous surface area, small surface area, if they've got the same number of transport proteins, it's less of an effect. Okay, however, in general, when you increase your surface area, you would increase your transport proteins. It was 10% of your surface area, it's 10%, it's still growing. Okay, so surface area is still affecting, but we've got this added level of what's our density of transport proteins, and on top of that, are they available? If it's a gated channel protein, is it open or closed? If it's a carrier protein, is it already moving things and it's not available for us? So we have that same sort of limitation as we do for enzymes when it comes to carrier proteins. When all of the carrier proteins are carrying, we can't carry any faster. We can't increase that diffusion any further. So, open, available. Open channel proteins, available carrier proteins, open, available, transport proteins. So we're adding many more layers into that concept of our diffusion to actually get a better idea of more levels of detail. Okay, so that then is facilitated diffusion. 3 more to go. Any questions at this point? Hopefully diffusion and facilitated diffusion, everybody's happy. Right, then we are sorted to talk about osmosis. So here again, we are doing passive transport. There's no energy involved anywhere in the system. Big deal, osmosis is only about water. It is movement of water only. It is movement from a high to a low water potential. You cannot say that water moves from a high concentration to a low concentration because it's like getting things confused. Concentration refers to solute. Yes, solute. The bits in there, okay? So we can't have highly concentrated water. That doesn't make sense, which is why we talk about water potential. So we measure concentration of water in terms of water potential. High water potential is close to 0. It means there's nothing in there. It's just H2O. That's it. Low water potential, we're going down on our negative scale. So minus 500, minus 1,000. Those have lower water potential because we've got lots of solute in there. It's not just H2O on its own anymore. So we're moving from high to low water potential. And the other thing that has to be involved for osmosis is across a semi-permeable membrane. So you've got 3 elements of this definition that have to be there, okay, that you have to think in mind when you're thinking about the process. It is movement of water from a high to a low water potential across a semi-permeable membrane. So here we come back to that story of diffusion. Can water diffuse? Yes, water can diffuse. But that is then movement from one dry area to one wet area to another dry area without the involvement of this membrane. So when you get to plant transport and you're talking about movement of water from one cell to the next, you have osmosis passing through the cytoplasm of one cell to the cytoplasm of the next, crossing the membrane. We have diffusion through the cell walls. So water can diffuse, but as soon as you cross a membrane, it is osmosis. So I've said here semi-permeable membrane. Partially permeable is exactly the same thing. We can use semi or partially interchangeably. So water, high to low, membrane involved. When it comes to osmosis, then you're going to have the same things affecting as diffusion. You have more surface area, then you get more osmosis, okay? And all of those things. So the thing we're going to focus on here is not animals, but animal cells and plant cells. Okay, so your little classic pictures, let's draw them here like this. Then for the plant cells, they would look. Remember, whenever you draw a plant cell, it has a double cell outside edge to indicate that cell wall. So even when you're scribbling, be in the habit of drawing your plant cells with 2 lines, wall and membrane, whereas your animal cells just have the 1 line for the membrane. Okay, and then we would have our plant cell would look like this. So, animal cells, it's straightforward. Okay, if you have water potential that is higher on the inside of the cell, then the water is going to move out and the cell is going to shrivel. So H2O out, cell shrivels. In this beautiful fried egg, let's draw a nucleus in for fun. We have no net movement of water. We are balanced potential inside and out. In this one, what's happened is your water potential outside the cell was higher, so water moved into the cell. And there is nothing stopping it moving in and in and in and in, and at some point your cell membrane can no longer hold all this water pressure, so your cell bursts. For plants, we get a little bit more technical because it is such an important part of plants. So we have plasmalized. Okay, when your cell membrane has pulled away from the wall, and now you can see 2 lines for the wall and 1 line for the membrane. Then these two, we have incipient plasmolysis. Where plasmolysis is about to happen, and we have turgid. And they look exactly the same. Okay, that's not a mistake. They look exactly the same. You cannot see when the cell membrane is about to pull away from the wall for incipient plasmolysis. You can only see once it has pulled away. You cannot see when the cell membrane is really stuck against the wall, when it's turgid. So they look the same, but it's 2 very different states. So pay attention when you're going through the 2 lessons for osmosis. Make sure that you've got all of this straight in your head. Okay, but that is the end of our 3 methods for passive. Diffusion, facilitated diffusion, osmosis. So now let's have a chat about our active processes. So let's talk about active transport. So here it's really important. We keep saying that it goes higher to lower, higher to lower, higher to lower. Concentration or water potential here is back to front. Here we are going from lower to higher concentration. We must note that this involves a carrier protein. And we must also note that ATP is involved. It is used. We cannot move up a hill without expending energy. You cannot roll a ball up a hill. You have to push the ball up the hill. So it's the same thing here. We cannot move from a low to a high concentration without expending energy. This is with regards to molecules and ions of all sorts of different sizes, okay? Because in this case, it's about moving from the low to the high. It's not random anymore, okay? So our specialized carrier proteins are called pumps. And they pump things into or out of the cell. So you'll have some pumps that work into, some pumps that work out of. Remember, different pumps for different functions, different pumps for different molecules. And the level of variety is enormous. We're only studying the sort of general concept. A nice example of active transport is when you get into plant transport, when you're looking at translocation, and it's the hydrogen ions that are actively pumped out of the cell. Okay, so hydrogen ions go through active transport to go out of the cell, then they want to diffuse back into the cell. We need facilitated diffusion to get them back into the cell. So it's really nice once you get there to give context to active transport and facilitated diffusion sort of in real life. But I'm running short on time, so let me stop digressing. Okay, in this case, ATP is required for your shape change of your protein. Fancy words? Conformational change. Okay, so facilitated diffusion, it just was the binding that caused it. Here, we need ATP to get that change of shape. That's active transport. So it's not complex, okay? But it is key things that you have to remember. Lower to higher through carrier proteins using ATP. Your last one is your bulk transport. Okay, so here you are moving large quantities or sizes. So for example, you are moving an entire bacterium rather than a molecule. Okay, so a bacterium is made of many, many, many molecules. So we need a different way to get him across the membrane. Okay, and again, ATP is involved in this process. It's an active process. So if we look at things coming in, we have endocytosis. This we split into phagocytosis and pinocytosis. So you've come across phagocytosis because you come across it in immunity at IG. Okay, we also have the exocytosis, which is the removal of things, the release of hormones, for example. When it comes to endocytosis, you've got a few important stages. You have a receptor on the membrane, and you bind to that receptor. You have ATP involved, and you have then membrane changes shape. So it's not now a protein itself is changing shape, it's the whole membrane that is invaginating in order to get a hold of this bacterium, for example. However, it is important to remember that the cell membrane is always intact. So your phospholipids and your proteins are moving around fluidly to maintain an intact membrane. So whether we're pinching off a new vesicle where the bacterium inside for phagocytosis, or whether we're fusing with an existing vesicle for exocytosis, that cell membrane always stays intact. Okay, so we've now been through all of them: diffusion, facilitated diffusion, osmosis, active transport, bulk transport. So make sure as you're going through that you get the details. And then when you come to the end of the unit, do yourself a nice little table highlighting similarities and differences, because that's the sort of thing that really helps to make things solidify in your mind.